Hello everyone. So today I will be briefly explaining some of the major proteins and enzymes which are involved in the DNA replication. So what is the DNA replication? It's one of the important processes that involves the synthesis of the two new daughter DNA from one single parent DNA with the help of the enzyme called DNA polymerase. Now DNA polymerase is one important enzyme of the DNA replication process that polymerizes the nucleotides together to synthesize a strand of the nucleic acid that is DNA. And this enzyme uses both the strands of the parent DNA as the template strands for synthesizing the new DNA. Here in this diagram, you can see that we have one parent DNA, one single parent DNA, and, and both of the strands of this DNA acts as the template strands, and using the enzyme DNA polymerase on this template strand, new DNA strand is synthesized. So this results in the formation of the two new daughter DNA. So let's now quickly revise the keynote features of the DNA replication process. One of the important features of the DNA replication process is that it is template dependent. Now, what is meant by this is that that when the enzyme DNA polymerase is adding the nucleotides into a new nucleic acid strand, so the information regarding the sequence of nitrogenous bases present in the new DNA is derived from the sequence of bases which is already present in the old DNA strand. And why is it so? So because the two strands, the old strand and the new strand, are complementary in nature. So that means if we have an adenine residue in the old strand, then at the same corresponding position on the new strand, you have a thymine residue, vice versa. And similarly, if in the old strand we have a cytosine, uh, then at the corresponding position on the new strand, you have a guanine residue and vice versa. So because of the complementary nature of the two strands, we can predict uh, the, the sequence of the new strand if we know the sequence of bases of the parent strand. So this information regarding the sequence of bases is present in the old DNA strand, and that's why it is acting as a template strand, which is absolutely required for the process to start. Now, second keynote feature is that this process is a primer dependent. So this means, what's a primer? Primer is a oligonucleotide. It's a stretch of the nucleic acid strand, which is like 10 to 12 nucleotides long. And onto this primer, that our DNA polymerase enzyme adds the nucleotides one by one by extending the 3' OH group of the primer. So one by one, it is adding the nucleotides with the formation of the phosphodiester bond between the nucleotides, and it is adding the new nucleotide at the 3' end of the newly synthesizing strand. So without the presence of a primer, the DNA polymerase cannot start building this DNA strand. So its presence is absolutely required. And this primer is made up of the RNA nucleotide and not the DNA nucleotide. This is the reason why when the replication process is being done, the RNA primers are removed and the DNA nucleotides are added uh, uh, in this gap which is created by the removal of the RNA primer. So once the replication is complete, it is being done, so the RNA primers are actually replaced by the DNA nucleotides. Well, another feature is that it's a bidirectional process. So this means that once the replication process is initiated, it leads to the creation of this replication bubble or replication fork. So this bubble or replication fork arises because it's created because of the unwinding of the DNA helix. Because we understand that in order for the replication process to start, the strands which are twisted together, they needs to be separated. Otherwise, the process cannot begin. All right. So basically, unwinding is done. We understand this unwinding is done by the helicase enzyme. There's more to it. We'll understand in the detailed mechanism. But let's just understand and focus on the bidirectional point. So this means that when this bubble, this pulse is created, this starts moving, this unwinding of the strands. This starts moving in both the directions. Both the directions means if it's a linear DNA, it is moving in the rightward and the leftward direction. And if it's a circular DNA, it is moving in both clockwise and anti-clockwise direction. So one of the other important key feature of the DNA replication process is that it's a semi-conservative process. And what is meant by this is that, like I said in the definition of DNA replication process, that it always results in the formation of the two new daughter DNA molecules, starting with a one single parent DNA molecule. So in this new daughter DNA molecule, one of the strand is the old strand, which is actually derived from the parent DNA, and the other strand is a newly synthesized strand, which is made by the DNA polymerase enzyme. So because one of the strand is old and the other strand is new in the DNA molecule, so that's why the process is referred as semi-conservative process. Now, another keynote feature is that this process is a semi-discontinuous process. Now, this means semi-discontinuous means that now one of the strand is being synthesized continuously, right? The the template strand which is running in 3' to 5' direction. For this template strand, the new strand is synthesized continuously in the 5' to 3' direction, which is the direction of the movement of the replication fork. But in case of the other template strand, okay, in the other template strand, we have this 5' to 3' direction. If you check the direction of the replication fork, the another template is in the is running in the direction 5' to 3'. And as we know, the new DNA strand can only be synthesized in one direction, that is 5' to 3' direction. So what happens in this template strand? What's happening is that the new DNA strand can only be synthesized, or only a possible way for the synthesis of for this template strand is that for this template strand, the new strand is being synthesized because it has to be synthesized in 5.3' direction. So it is being synthesized in the direction which is opposite to the movement of the replication fork, right? So what we see is that one of the template strand, the new strand is being synthesized continuously, right? But for the another strand, this is being synthesized not continuously but discontinuously. Discontinuously means it is being synthesized in fragments, right? Because as in when the DNA is like unwinded again and again over the time, so that only that part, only that stretch is being synthesized. It is being synthesized in the direction opposite to the replication fork. So because one of the strand is continuously synthesized and the strand is synthesized discontinuously, that's why the process is referred to as a semi-discontinuous process. Another important key feature of PNR application is the presence of the Okazaki fragments. Now, like we discussed earlier, now one of the strand, which is the template strand, which is running in 3' to 5' direction. For this strand, the new DNA strand is being synthesized continuously. But for the other template strand, which is in the direction 5'3 prime, for this template strand, the new DNA strand is being synthesized discontinuously, and it is called as the lagging strand. So lagging strand is the one which is being synthesized discontinuously, and it is being made in fragments, and these fragments are referred to as the Okazaki fragments. So in case of prokaryotes, these are like Okazaki, the size of Okazaki fragments are between 1,000 and 2,000, but in case of eukaryotes, the size is much smaller in the range like 100 to 200 nucleotides. Another feature is the presence of origin of replication. This is the site. This is the specific site where the replication process starts or begins. So that means this is the site where initially the separation or the binding of the DNA helix, DNA strands takes place. Another important feature is that DNA synthesis always takes place in a particular direction, and this direction is the 5' to 3' direction. And why is it so? Because we have a hydroxyl group attached at the 3' site, and this OH group is the main site. The 3' OH group is the main site where the new nucleotides are being added one by one by the DNA polymerase enzyme. Now let's quickly discuss some of the major enzymes and proteins involved in the DNA replication process. Now, one of the main replicative enzymes of the DNA replication process is the DNA polymerase enzyme. Now, as the term itself explains, polymerases are the enzymes which polymerizes or adds the nucleotides together into a new strand of nucleic acid or into a new strand of DNA. So what it is doing is that it is adding the nucleotides one by one with the formation of phosphodiester bond between them and polymerizing the monomeric units into a strand of nucleic acid. We have different types of DNA polymerases present in prokaryotic cell and eukaryotic cell. So let's first discuss the different types of DNA polymerases which are present in the prokaryotic cell. Well, mainly there are five types of DNA polymerases: DNA polymerase 1, 2, 3, 4, and 5. We'll discuss DNA polymerase 1, 2, and 3 here, and DNA polymerase 4 and 5. It's a complex, and it's mainly involved in the DNA repair synthesis. It's an enzyme which is used to repair the DNA mutations. So it's a repair enzyme. So one by one, we'll discuss these three types: DNA polymerase 1, 2, and 3. Now, DNA polymerase 1 is involved in the DNA replication process. It has a 5' to 3' polymerization activity. As such, you will see that these polymerases do have a 5' to 3' polymerase. This is the main activity, means they are adding the nucleotides in 5' to 3' direction. It is the direction of synthesis of the DNA strand, as we know already. And it also has a 3' to 5' exonuclease activity, as well as 5' to 3' exonuclease activity. Now, these two activities are important for its function. So because it has this 3' to 5' exonuclease activity, it can proofread the DNA. Means if in case there is a presence of a mismatch, not complementary base pair, so it can remove that non-complementary base and replace it with the correct base pair. So it can proofread the DNA and is involved in the repair synthesis. And also one of the important activities that it has a 5' to 3' exonuclease activity. Now, because of this activity, it has the ability to remove the RNA primers from the DNA strand, and the gap which is thus created because the removal of the RNA primer, it can fill it, replace the RNA primer with DNA nucleotide. So that means it can remove the primer, it can fill the gap, and it can uh, seal the leaks. That's its activity. Now, DNA polymerase 2, it has a 5' to 3' polymerase activity, as well as it has a 3' to 5' activity, and as such, it is also involved in the repair synthesis. Now, one of the main type of DNA polymerase, which is majorly involved in the synthesis of the new DNA strand, is the DNA polymerase 3. So this is the main replicative enzyme which is present in the prokaryotic cells. It is quite large. It's a large complex made up of at least 10 different subunits. And of course, it has a 5' to 3' polymerization activity because it's the main replicative enzyme. And it also has a 3' to 5' exonuclease activities, which gives it the ability to cleave the DNA. So this is the main enzyme which is mostly synthesizing the new DNA strand during the replication process. Now, in case of eukaryotic cells, there are more than 10 to 15 different types of DNA polymerases, and there are different types. But here we are discussing the important ones, like there is DNA polymerase alpha, beta, gamma, delta, and epsilon. So DNA polymerase alpha has a primase activity. It has a primase activity. That means it has a polymerization activity. It polymerizes the DNA nucleotides in 5' to 3' direction, but it has no exonuclease activity. It can't remove the DNA nucleotide. So DNA polymerase alpha, we see that it's able to make the primer during the during the DNA replication process. When it starts, it makes the RNA primers, which is required for the replication process to start. And also it kills during the annihilation phase. Not only it makes the primer, it also gives a little bit of extension to the RNA primer, which is then the activity is then taken over by the main DNA polymerase enzyme, which is delta or epsilon. Another DNA polymerase type is the beta type. As such, beta type is mainly involved, and we'll not discuss it much in detail. It is involved in the DNA repair synthesis, like in the base excision repair and nucleotide excision repair, and so it's mainly involved in the repair of the DNA mutation. Another important DNA polymerase type present in the eukaryotic cell is the DNA polymerase gamma. This is a type of DNA polymerase which is involved in replicating the mitochondrial DNA. So this is not in the nucleus. This is in the mitochondria, and it is replicating the mitochondrial DNA, and it has no 3' to 5' exonuclease activity. Another two types, delta and epsilon, these are the important types. Now, delta is responsible for the synthesis of the ligase strand during the DNA replication, and it has 3' to 5' mechanistic activity. And another important DNA polymerase is the epsilon DNA polymerase. It's synthesizing the leading strand during DNA replication, and it also has a 3' to 5' exonuclease activity. So another set of enzymes which are present in the DNA replication process are the DNA helicases. Helicases are important set of enzymes because these are the enzymes which are responsible for separating or unwinding the DNA strands. Because until and unless the DNA strands are separated, the new daughter DNA strands cannot be synthesized, cannot be elongated. So if new DNA strand has to be synthesized, it is required that the DNA helix is continuously separated, and the strands are separated with the help of DNA helicase enzyme. It is separating the DNA strands by breaking the hydrogen bond which exists between the strands of DNA, and the energy which is required for this is derived by the hydrolysis of the ATP. So as such, it also has a ATPase activity in it, so that it can obtain this ATP energy and use it to break the hydrogen bond in order to separate the DNA strands. Another enzyme is the single-stranded binding protein. So as the strands are being separated, we know that these are complementary in nature, and as such they have a tendency to renature again. Uh, so they have a tendency to again form the hydrogen bonds. So in order to prevent the re-annealing of the separated uh, strands, this SSB protein or single-stranded binding proteins, they coat both of these strands so that until this process is complete, the replication of the strand is going on. These strands are not re-annealed to 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 create the helix again. So this is prevented with the help of this SSB protein. Another enzymes which are important are the topoisomerase enzyme. Now, topoisomerase enzyme are important. Why so? Because as the replication fork is moving, we understand, we already know that DNA has a helix. Okay, these are the two strands are coiled with each other. So as the replication forks are moving, so what happens is that the number of twists, the coiling increases ahead of the replication fork, because at this replication fork, what is happening? The strands are being separated, right? But ahead of this replication fork, what is happening? Supercoiling is taking place. We have a higher number of coiling. The twist number is increasing. So because of this, if this like the if this coiling is not reduced, so it will be impossible for the replication fork to continue moving. So in order to reduce the supercoiling in the ahead of the replication fork, topoisomerase breaks this phosphodiester bond in a DNA strand, and it untwists the DNA in order to reduce the supercoiling, so that replication fork can continue to move and synthesize the new DNA strand. So this is done by the topoisomerase enzyme. So another enzyme is the DNA gyrase. Now DNA gyrase is a type of a topoisomerase. Is a type of a topoisomerase which is again uh, used to reduce the supercoiling which occurs ahead of the replication fork. Other important proteins are the beta-clam proteins and the DNA ligase enzyme. Now, beta-clam proteins and its corresponding protein in eukaryotes is called as the pCNA protein. Now, this beta-clam protein or pCNA protein are actually the proteins which are they have a ring-like structure, and they binds our main replicative enzyme as well as the other proteins of the DNA replication process. And these are binding these enzymes together so that these are being prevented from leaving the strand, so they are not dissociated easily. And also, because these beta clam proteins, they have a ring-like structure, so they actually are sliding over the DNA strand. they are loaded onto the on this DNA molecule, and these are sliding over between the strands. So what it does is that it increases the rate of polymerization of the DNA synthesis. It is increasing the rate of DNA synthesis because instead of moving now the replicative enzymes and other proteins, they are sliding. They are associated with the beta gland protein, and beta gland protein, it is sliding on the DNA strand. So the rate of polymerization, the rate of DNA synthesis is drastically increased. So another important enzyme is the DNA ligase. DNA ligase is an enzyme which joins the two DNA fragments together. So its main purpose is to join the DNA fragments together. For example, it have in the DNA replication process, it helps in joining the Okazaki fragments together, because we know that the the lagging strand is being synthesized discontinuously in fragments called as Okazaki fragments. So when the replication process is done, these Okazaki fragments are then joined together to form a one continuous strand. So this is done with the help of a DNA ligase enzyme. Thank you so much for watching the presentation, and hope it benefited you in whatever little way it could.